So in this second lecture on immune metabolism, we're going to focus on lymphocyte metabolism in particular. So I'll remind you, in the last lecture we discussed how metabolism can support immune cell function, the different roles it can play in generating energy, in supporting biosynthesis, and all the different fuels that can feed into these processes. We also mentioned how this is particularly dramatic and important in immunology, because immune cells change their functions so much and so dramatically upon immune activation. So there are very clear changes in metabolism that are associated with immune activation. So today I'm going to tell you about the metabolism of three types of a lymphocyte: natural killer cells, which we've worked on quite a bit, T cells and B cells. Now, we can't cover all the lymphocytes in this lecture, because just for time constraints, and I guess also because some of the information on other lymphocyte populations, some of the less abundant ones in the body, are less well developed. So we know less about metabolism of, for instance, innate lymphoid cells. So that's why we're only going to talk about natural killer cells. There's also an emerging understanding of the metabolism of other different types of T cell subsets, but we're going to talk mainly about the alpha beta T cells, though there is some nice work from Lydia Lynch that's just been published on gamma delta T cells, which you can check out if you're interested. But for the interest of time, we can't really focus on all that, but maybe future years we'll start to incorporate that. But today I'm going to focus on natural killer cells, T cells and B cells, and tell you a little about the metabolic configurations, the key metabolic regulators that control their metabolism, and how this ultimately supports their unique functions, differentiation and functions. So I'm going to start off with natural killer cells, which my lab has worked on for the last 9 or 10 years. So natural killer cells are lymphocytes. They're classically known as part of the innate immune system. So they are primed and they're ready to go. So they can recognize target cells and kill them straight away. They can encounter cytokines and make interferon gamma and other cytokines straight away. So they are ready to exert their functions immediately, and that's a key part of the innate immune system, is that they are fast acting. So for instance, they respond to innate cytokines like IL-12, 18, 15. They make key effector cytokines like interferon gamma, TNF alpha. They also make important chemokines, and they can directly kill target cells, whether they be virally infected cells or tumor cells. But that's not the whole sum of their function. They can be activated for longer time periods with cytokines, and so they can work immediately, but they can also act and function for longer time periods, for hours and even days. And certainly when they're activated with cytokines for the 18 to 24 hour time point, they have increased function. So while they are already able to kill, if you stimulate them for a long time, they become better at killing, and they're able to make more interferon gamma. And in fact, they are also able to work alongside the adaptive immune response. So once the T cells become activated, they make cytokines like IL-2, and IL-2 is a potent stimulus for NK cells, in particular for NK cell metabolic responses. So really, I think NK cells are considered now to be a lymphocyte subset that sort of bridges the innate and the adaptive immune response. So we're going to think about some of these metabolic changes that happen over the time period of 12 to 24 hours. I should say that the really immediate functions like direct killing and making interferon gamma immediately following stimulus, they aren't associated with metabolic changes, so they're ready to go with that. But these enhanced functions over time, they're linked to clear and robust metabolic rearrangements within the NK cell. So I'm going to tell you a little bit about what we know, all summarized on this slide. So in the most part, we have studied the detailed metabolism using cytokine stimulation IL-2 plus IL-12. But actually, while we haven't done as much detail with other cytokines, we see very similar patterns when we stimulate just with IL-15, high dose IL-15, or when we stimulate with doses like 2, 12, and 18, or even 15, 12, and 18, which are all important combinations. 15, 12, and 18 is the cytokine stimulus that is used for adaptive transfer immunotherapy using NK cells. So, in general, the resting NK cell before a stimulus has relatively low levels of cellular metabolism, uses relatively small amounts of glucose. But when you activate with cytokine for 18 hours, you get a robust increase in glucose uptake. So they start taking up lots more fuel, and they metabolize most of this to lactate. I think I mentioned this in the last lecture. So if we do some metabolomics using traced C13 labeled glucose, so you'll remember from my lecture on metabolomics earlier in the year, what tracing metabolomics is when you add, instead of carbon 12, you add carbon 13 to the fuel that you're adding, and you can trace where those carbons with an increased mass go using the very sensitive mass spectrometry. And so when we did this using heavy labeled glucose, we could find that almost 80% of all the carbons that are given to the cell in the form of glucose end up in lactate and are secreted from the cell when we stimulate with IL-2/12. But there's also, this glucose also feeds into the mitochondria, and we have high levels of OXPHOS, so we can, a good readout of OXPHOS, as you will know from our last lecture, is oxygen consumption. We can use this machine, the Seahorse extracellular flux analyzer. I think you had lectures on this earlier on in the year from Richie Porter. So here is the oxygen consumption rate of unstimulated and stimulated NK cells, and you can see you get a robust increase in oxygen consumption, and you'll know what all these different inhibitors are from Richie's lecture. This gives you these difference here. It tells you the HB linked respiration. This gives you the maximum respiration, etc. But the general point here is that they have increased OXPHOS when you stimulate them with cytokine. What about some of these other sort of other pathways that we discussed in the last lecture, like the pentose phosphate pathway, which is important for biosynthesis, important for making NADPH to sustain your reduced levels of glutathione, so you can maintain redox balance. But we've also looked at this, and to do this, we did some more metabolomics using heavy labeled glucose. But in this case, we used 1,2 labeled glucose, so only carbon 1 and 2 has the heavy labeled carbon, and carbon 13. And what this allows you to do is to measure the lactate at the end of it and determine what route the glucose takes. So whether the glucose goes straight through canonical glycolysis to make lactate, or whether it gets passed through the pentose phosphate pathway and back into glycolysis. And this would tell you whether they are prioritizing the production of NADPH. And you'll remember we discussed this in the last lecture about how the pentose phosphate pathway can be used to make to prioritize the production of ribulose 5-phosphate for making nucleotides, or it can be tuned to focus on making NADPH. So the reason this works is because depending on which route the glucose take, you get a slightly different pattern of label incorporation into lactate. So here's your 3-carbon lactate. If it goes through the glycolysis, you only get 1,2 labeled lactate, whereas if the glucose goes through the pentose phosphate pathway, you get this 1,3 labeled lactate. And the nature of mass spectrometry is when you ionize this, you lose the carboxylic group. So you end up with 2 carbons being measured in the mass spectrometer, and if it's come through glycolysis, it's a heavy labeled 2+, so extra 2 atomic weight. Whereas if it goes through the pentose phosphate, you only have 1 of these heavy labeled carbons, so you can distinguish which route they went through. A long-winded explanation, and here's the simple result at the end of it. When you activate NK cells, you get this robust induction of glucose going through glycolysis, but you also get a significant increase in the amount of glucose going through the pentose phosphate pathway, showing that these cells are engaging all these metabolic pathways to meet the demands of NK cells, and suggesting that one of the important functions in these activated cells is dealing with redox stress, dealing with reactive oxygen species. And certainly, if you look at reactive oxygen species, so let's just remind you. So I'll come back to this now. So I'm getting a bit ahead of myself. I'll come back to this now in a minute. So we will also discuss the fact that in NK cells they're quite interesting because they don't use the TCA cycle. They use the citric malate shuttle, which I'll describe later. But nonetheless, they still have high levels of OXPHOS. So remember, this is the pentose phosphate pathway, as we said. And just reminding you that it's a branch of glycolysis, and it's important for dealing with reactive oxygen species. And as I said, we measured reactive oxygen species in the NK cells, and certainly when you activate with cytokine, you get this clear increase in reactive oxygen species being produced within the cell. So this makes sense why you might be diverting glucose through this pentose phosphate pathway to generate your NADPH to allow you to maintain redox balance within the cells. So as I said, one of the interesting features of NK cell metabolism is that they don't use the TCA cycle to a great extent, unlike a lot of other cells, but actually engage what's called a citric malate shuttle, which is a cycle of sorts as well, but that crosses the mitochondrial membrane. So I'm going to give you a bit more detail about how this works and what the outputs of this cycle are compared to the TCA cycle. So acetyl-CoA for the citric malate shuttle is used to create citrate, which is the same as the first step of the TCA cycle. In the TCA cycle, this citrate is then further metabolized down through a range of intermediates, including alpha-ketoglutarate, succinate, fumarate, malate, oxaloacetate, and goes round and round that way. But in the case of the citric malate shuttle, the citrate is exported from the mitochondria through SLC25A1, a transporter. It is metabolized in the cytosol into malate, which comes back in through SLC25A1. It's then further metabolized to oxaloacetate and then combines with another acetyl-CoA to give you citrate. So this is a cycle as well, and the outputs of this cycle, the citric malate shuttle, are reducing equivalents. So you get per glucose, you get 2 NADH, which can feed into the TCA to the electron transport chain, generate this proton gradient, and drive ATP synthesis. But you also allow for the restoration of NAD+. So here, conversion of oxaloacetate to malate uses NADH and generates an NAD+. And as we said in the first lecture, NAD+ is an important cofactor for GAPDH. So this is simultaneously supporting glycolysis by regenerating NAD+, and generating the reducing equivalence that feeds electrons into the electron transport chain for driving OXPHOS. So, are there any other outputs of this cycle? So, another key output that's different from the TCA cycle is the fact that you get cytosolic acetyl-CoA, which might be important for lipid synthesis or acetylation reactions. Now, in our NK cells, we think this is, although we still need to work on this, we think that this acetyl-CoA is important for acetylation reactions, and that could be epigenetic changes. But acetylation can happen on a lot of different proteins and control protein function. So for some reason, which is yet to be determined, NK cells are prioritizing acetylation through using this shuttle. So there's a basic signature of the NK cell metabolic sort of configuration. And this configuration allows you to make energy. So the high flux to glycolysis is inefficient to making energy, but still the high flux means you're still making a fair bit, a good amount of energy, and then you're also generating more efficient ATP through oxyphosphorylation. As well as this, you're supporting biosynthesis. The high flux to these pathways allows you to generate amino acids such as serine and alanine to generate acetyl-CoA for biosynthesis. And also through the pentose phosphate pathway to make NADPH, which is that important cofactor for biosynthesis. This also supports acetylation reactions, and of course the NADPH can also be used to help to control redox status within the cell and deal with reactive oxygen species, as we've already discussed. So alongside these changes within metabolism, so what's required? Well, there's a distinct increase in the metabolic machinery that supports this. So we've increased expression of transporters, like for glucose, glycolytic enzymes. We also have increased mitochondrial mass, which allows for this increased oxphos. So there's clearly increased flux, and there's also increased machinery to deal with, to facilitate this increased flux. So what are the key sort of regulators that are required for this? What are the key metabolic signaling pathways that support this metabolic configuration? Well, as I said, one of the most interesting aspects of NK cell metabolism is the fact that they use the citrate malate shuttle. So what allows them to switch to this? Well, it's this transcription factor here, sterile regulatory element binding protein, SREBs. Because these two key components of the citrate malate shuttle are SREB target genes. This transporter here, SLC25A1, and ATP citrate lyase here. So this is unusual and unexpected because sterile SREBs are known to be master regulators of fatty acid synthesis and cholesterol synthesis, and they are not known to regulate glycolysis and oxphos until this current study. So it was a nice unexpected surprise. So in general, they control fatty acid synthesis, they control cholesterol synthesis. They're quite a complex mode of activation, which we won't go into today. But they're interesting when this does become important. They are inhibited by cholesterol and also by other oxidized cholesterol species like 25-hydroxycholesterol, 27-hydroxycholesterol. So that's important because that has implications for NK cells working within inflammatory environments or within tumors. So SREB in this situation is a key regulator of glycolysis and oxphos. And if you inhibit SREB, the rates of glycolysis and oxphos are dramatically reduced. And then another key transcription factor is c-MYC. c-MYC was less of a surprise because this is known to be important in controlling growth, and it's often overexpressed in cancer. And this induces the expression of glycolytic genes and glucose transporters in NK cells, and also stimulates mitogenesis, which is the production of new mitochondria. Again, it's a complex mode of activation, but one key thing to know is that c-MYC abundance drops rapidly when amino acid levels are low. So this means that the NK cells are quite sensitive to amino acid deprivation, in particular glutamine deprivation actually. So the SREB seems to be controlling the configuration, whereas the c-MYC is controlling the machinery. And I should mention here mTOR, which is a kinase complex, which is also very important, known to be very important metabolic regulator, also plays complex roles here, so which we won't go into the absolute details of. But suffice to say that it's important. mTOR can regulate maximal SREB activity. It also, at certain time points, it can control c-MYC activation. So is this metabolic configuration important for NK cell responses? And the answer to this question is a resounding yes. So this slide summarizes a huge amount of work by ourselves and others, showing that inhibitors of these metabolic pathways directly, either glycolysis or oxphos, inhibitors of these different metabolic regulators, knockout mice of these different metabolic regulators. These are all work that we've done. We've looked at inhibitors of mTOR. Others have looked at knockout models for mTOR. No matter what you do, targeting any of these aspects of NK cell metabolism results in a clear and quite a substantial decrease in NK cell functionality in terms of their cytokine production and their ability to kill target cells. So here's just one little piece of data to confirm that. So if we use an inhibitor of SREB, 25-hydroxycholesterol, and look at the ability of these cells to kill target cells. So this is just a cytotoxicity assay where we put NK cells with tumor cells at different ratios, and we have the tumor cells loaded with a fluorescent dye. So when they're killed, they release the dye, and it's very easy to measure specific lysis. And here you can see the NK cells are very good at killing when they're activated with cytokine. At this ratio, they're killing almost 8 out of 10 of the tumor cells. But if you activate them in the presence of this SHREB inhibitor, their ability to kill these tumor cells is dramatically reduced. So somehow SHREB is really important for this process of recognizing the target cell, polarizing the cytolytic granules that the NK cells have, and to killing the target cell. And we're now looking at, one of the students in my lab is now looking at exactly what part of this process is inhibited by oxysterols to inhibit the ability of NK cells to kill tumor target cells. We've also looked at this in a model of NK cell cancer immunotherapy. So this is a model where we give mice a melanoma, B16 melanoma cells. So it's a model of melanoma in mice. We give them the tumor cells on day 0, and then we give them 3 therapeutic injections of NK cells that we have expanded ex vivo and then stimulated with a cytokine combination IL-12, 15, and 18, and then we measure the tumors. And it's very clear that when you give these activated NK cells, they have a therapeutic effect here in the blue line compared to if you just inject PBS. So these NK cells are working to kill the tumor cells and reduce tumor growth. But if we activate the NK cells in the presence of 25-hydroxycholesterol, we completely lose this therapeutic effect. So again, showing you that NK cell metabolism is essential for the therapeutic activity of these NK cells in this cancer immunotherapy model. And here's just the final tumor volume on day 15 for each of the conditions. So when we think about cancer immunotherapy, I think about, basically the aim is to kickstart the immune system to start to kill the tumor again. Our immune systems have the ability to do this, but when we have cancer, often our immune systems have stopped doing this for whatever reason. So we need to find a way to restart the patient's immune system to kill the tumors. So I think about how to start up and drive a car. This is a very, very simple analogy, but hopefully I think it's something quite useful. So if you want a car to move forward, you need to, first of all, take the brakes off. So we need to think of what are the brakes on the immune system that are affecting their ability to recognize and kill cancer. You need to put the foot down. So obviously, unless your car is parked on a hill, taking the brake off in itself is not going to do anything. You need to, put the foot in the accelerator. But even then, putting your foot in the accelerator will do nothing if the engine is not turned on, or if there's no fuel in the tank. So when I think about your NK cells and using NK cells for cancer immunotherapy, what does this mean? How does this translate to think about NK cells? Well, so in terms of the brake, we need to think about what inhibitor receptors are being engaged on the NK cells. So for T cells, we know that the PD-1 is an important inhibitor receptor, and that's the nature of why checkpoint inhibitors work. PD-1 can be expressed in NK cells, but there are other inhibitor receptors that can also be expressed on NK cells, and that could be the reason why they're not working in cancer patients. We need to think about what the right stimulus is to stimulate NK cells to exert their anti-tumor functions. But our work is more thinking about the engine. So we need to think about what the metabolic signaling that the NK cells need to have to make sure that the engine is running. And we need to think about nutrient availability. So when they're in the tumor, is there nutrients available? We know that glucose is important here. So what we need to think about for NK cell immunotherapies is whether we can sustain key metabolic signaling, mTOR, c-MYC, and SHREB when those NK cells are in a patient trying to kill a tumor, and whether we can sustain availability of glucose. Availability of amino acids are also important for NK cell responses. So I've talked about this in terms of NK cell responses, but it's also very true for T cell therapies as well, and for cellular immunotherapies to viruses and other infections as well. So these are all the key considerations if we're trying to drive an immune response. Obviously, if we're trying to inhibit an immune response, it's completely different considerations. Okay, so hopefully I think I've given you a good picture of NK cell metabolism, some of the key regulators, and how we can start thinking about using that information to design better therapies. And in particular, our focus in the lab is for designing better therapies against cancer. But all those sort of metabolic, we also do work on some using some viral models, and the same thing is true if we during viral infection. NK cell metabolism is also very, very important. So as always, I tend to talk about NK cells a bit too much, as it's something we work on, but we really must move on now to talk about our second lymphocyte population, T cells. Now, I worked on T cells a long time ago now, during my postdoc with Doran Cantrell in Dundee. So I worked on CD8 T cells, and so I know a bit about these. CD8 T cells, when activated, they can have important functions again in antiviral immunity and anti-tumor immunity. They can differentiate into different subsets. So they differentiate into these cytotoxic effector T cell that can recognize and kill target cells. But as with the adaptive immunity, you also generate memory cells. And these are long-lived cells that give you immunological memory to help you deal with subsequent infections or with similar viruses, and allow you to deal with them better the second time. Similarly, CD4 T cells, the key subset of alpha and beta T cells, can differentiate into different types of immune cells. They can differentiate into Tregs, which are important in keeping the immune system in check, and they can differentiate into a range of different effector T cell subsets. On this slide I just mentioned Th1s and Th17s, but there's also Th2s as well, so which have different roles and different types of immune responses. So what are the metabolic configurations of these cells as they go through these responses and form these different subsets? So when you activate a CD8 T cell through the TCRs and co-stimulation, so this is initial immune triggering of T cells, you get a robust increase in glycolysis. And in these cytotoxic T cells, these high levels of glycolysis are sustained over time until the end of the immune response, when the T cell numbers collapse through apoptosis. OXPHOS is also increased, so they really ramp up both metabolic pathways, quite similar to in a way to NK cells, and these are sustained over the course of the immune response, and this immune response is kept going by the cytokine IL-2. So we think about the CD8 cytotoxic T cell response being a combination of aerobic glycolysis and high levels of OXPHOS. Now, to generate a memory T cell is different. You initially get the same burst of metabolism driven by the TCR, but you then get a reduction in the glycolytic rate, but you sustain the increased levels of OXPHOS. So these cells are anaerobic glycolysis plus OXPHOS initially, and then they turn into these cells that are prioritizing oxidative phosphorylation. It's quite distinct patterns of metabolic changes as they go through the lifetime of these differentiation steps. Now, c-Myc is important in this initial triggering event where glycolysis and OXPHOS is increased. And then in your effector T cells, HIF1 alpha is important for the sustained glycolysis, this IL-2 dependent sustained glycolysis in these effector cytotoxic T cells. And mTOR is also important here in differentiation between your effectors and your memory cells. Your effectors maintain high levels of mTOR, whereas your memory cells, it's important that they downregulate mTOR at this point here to generate your memory T cells and to change your metabolism appropriately. So, as I said before, when we think in general of effector T cells, and this is probably more true for both CD4s and CD8s, although this is a kind of combination of what we know for both. Effector cells have high levels of glucose uptake, they are engaging aerobic glycolysis, whereas these memory cells have more complicated metabolism. They are able to generate fuel stores, they're more oxidative in general, aren't really engaging in aerobic glycolysis to the same extent. So very distinct metabolic outcomes that correlate with very distinct sort of differentiation outcomes of memory cell versus effector cell. So another key observation is that the cytotoxic T cells have very different mitochondrial configuration. So here is your mitochondria. My drawing of mitochondria in a cytotoxic T cell. They're small. They're relatively independent mitochondria. They also make lots of reactive oxygen species. Whereas in the memory cell, you have these nice tubular networks of mitochondria, which are much more efficient at OXPHOS, and they don't make reactive oxygen species. And of course, reactive oxygen species isn't really conducive to longevity because this causes damage within the cell. So you can see why this mitochondrial configuration will be more suitable for a cell that you want to live for a long time. So if you start playing around with the metabolism, can you affect the differentiation? Well, absolutely you can. So if you want to favor the differentiation of memory cells, you can inhibit mTOR using mTOR inhibitors. You can inhibit glycolysis, and that will also favor memory cell differentiation. You can also inhibit mitochondrial fission, so you can maintain the mitochondria, promote this network mitochondria, and that in itself can also seem to push towards memory cells. On the flip side, if you want to prioritize cytotoxic T cells, you take steps to activate mTOR. So if you knock out AMPK, which is an inhibitor of mTOR, you can favor cytotoxic T cells, and they can no longer make memory cells. And if you disrupt fatty acid oxidation, which is important for OXPHOS in these cells, again, you can't make memory cells, and so now all your T cells go to form cytotoxic T cells. So of course, these are different reasons why you might want to promote the differential differentiation. That's a bit of a mouthful. Why you might want to promote the differentiation of one subset over the other. These are important in viral and tumor responses, but they are also associated with some of the pathogenesis associated with immune diseases, and also in viral responses, they can also cause some of the damage through being overactivation and not being kept in check. So there are reasons why you might want to dampen down these cells in certain situations. There are also reasons why you might want to increase these cells, for instance, in the case of tumors. And of course, these memory cells are important because these are part of our immunological memory. And some of the vaccines some of the diseases we still haven't got good vaccinations for. One of the reasons behind this is because vaccination it's difficult to generate these memory T cells, and some of the diseases we're trying to generate vaccines for, like HIV, we really need this sort of cellular response, this cellular memory. Now, what about CD4 T cells? Well, it's a similar story. There's very different cellular metabolic configurations of the Tregs versus the CD4 effector T cells. The Tregs in particular, they scavenge fatty acids from the environment, and they use that as a fuel, which is different from some of the effector cells, like for instance Th17 cells. Go backwards. So again, there's in general, if you've low mTOR, you favor Tregs. If you've high mTOR, you favor effector T cells. So you can promote Treg differentiation by inhibiting mTOR, or also by inhibiting glycolysis. Whereas you can promote effector T cells through mechanisms that activate mTOR. So again, there's a key balance here between metabolic signaling, cellular metabolism, and CD4 T cell differentiation. So of course, these are the abnormal differentiation between effectors and Tregs is associated with certain diseases. So in cancer, you tend to have too many Tregs and not enough effector T cells, and that's a problem. In autoimmunity, you tend to have a lack of Tregs and too many effector T cells, which are causing damage. So you can see how manipulating the balance between these two cell subsets could be useful for treating these types of diseases. So for instance, if you want to treat autoimmunity, you might want to promote the differentiation of Tregs over Th17s. And a very elegant study took up this question, and they noticed that Tregs and Th17s differ. One of the key metabolic differences was the fact that Tregs burn fatty acids that they scavenge, whereas Th17s make their own fatty acids. So the fatty acids that the cells won't be using for making lipid membranes, for instance, they are scavenged by Tregs, whereas Th17s need to make their own fatty acids. So this group in this study decided, well, what happens if we inhibit de novo fatty acid synthesis? based on the slide I showed you previously, it seems like it's only Th17s that make their own fatty acids, so maybe they are the only ones that will be susceptible to an inhibitor of fatty acid synthesis. And that was exactly the case. So they used this inhibitor of the first step in fatty acid synthesis, acetyl-CoA carboxylase, called SORA, and when they added this, you can see there's a clear switch in the differentiation of the cells. So you can look at Th17s as cells that are making IL-17. Here you have lots of Th17s, but if you inhibit fatty acid synthesis, you promote lots of Tregs, and we measure Tregs by the expression of FOXP3. So a clear shift in the balance towards Tregs here in this setting. So they then said, well, how does this have a therapeutic benefit in autoimmune disease? And they used a mouse model of MS. And here you can see the induction of MS in normal mice. You can see that over a number of weeks you start to get increased severity scores. The black bars here are severe disease. Whereas when they treated the mice with this inhibitor of fatty acid synthesis, you can see that the onset of disease was severely delayed, and when it did come on, it came on less severe. Very interesting, showing that an inhibitor of metabolism could have a therapeutic benefit. Is there any other studies that looked at this? Well, early, a good few years ago now, when was this, back in 2015, Jonathan Powell's group asked whether, just adding a cocktail of just basic metabolic inhibitors could have an effect on T-cell responses. And they decided to do this in allograft models, mouse models, so transplantation models. And the reason that rejection of transplants is a T-cell driven event. So they said, well, how about at this stage in 2015, we started to know a bit about T-cell metabolism. So he said, what if I just feed the mice 2-deoxyglucose, metformin is given to patients. It's a very safe drug. 2-deoxyglucose has been shown to be safe in mice. And there's also this other inhibitor of glutamine metabolism, DOM. So they just fed these cocktail of metabolic inhibitors and looked at what happened to mice after they received the transplant. So first of all, I guess they did some in vitro studies looking at the differentiation of these different cells, and it was very clear that the differentiation was affected. And here's I'm just going to show you some of the data for the in vivo model. So this is a skin transplant model, which is quite nice because you can actually visualize it. So here they just transplant a patch of skin from a white mouse to a black mouse, and obviously in the absence of any treatment, the skin graft is rejected pretty quickly within 10 days, and you see the loss of the white skin and the white fur. But if you start adding in these inhibitors one at a time, or the triple therapy, here you can see the rejection is delayed by a long time, and now it's taking up to 40 days for the rejection of the skin transplant just by adding these three metabolic inhibitors. They also did a heart transplant model, and you can see if you transplant a heart from one strain of mouse to the other, obviously it's rejected very quickly, and the mice can't survive. But if you add these triple therapy, you get prolonged survival, which is great. Okay, so metabolic alterations, metabolic perturbations can affect differentiation, and we'll talk a bit more about functions in the next lecture. Okay, just one thing about the citric malic shuttle. I often get asked this: do T cells also engage in the citric malic shuttle? And my answer is usually nobody's really reported it because they haven't probably looked close enough. Now, certainly, this is the key experiment we use to show that they're engaged in the citric malic shuttle. We gave them heavy labeled glucose, and if you do that, as that goes into the mitochondria, you get a +2 labeled citrate. Now, if that goes into the TCA cycle, these heavy labels get fed down to subsequent metabolites like fumarate. But if it gets exported from the mitochondria, you don't get any heavy level in the fumarate, and that's exactly what we saw in NK cells. So here's N+2 citrate. So there's lots of it. The majority of the citrate is N+2, but very little of the fumarate is N+2. Now, if it was going around the TCA cycle, you get N+2 citrate, and you'd also get N+2 fumarate up here. So it was a very clear result. Now, if you look at some of the metabolomics data that's out there, and often not referenced to, even though this is in the supplemental material of a nice paper. I'll just read with the reference here. I can't remember what it is. Ming Li paper. But you can see, forget about the blue bars, that was a knockout mouse they're looking at. But just looking at the wild type mouse, you can see they did the same experiment, giving it heavy label glucose, and the majority of the citrate is M+2, and very little of it is converted to fumarate. So suggesting to us that they are probably exporting the citrate into the cytosol. But nobody's really looked in a huge amount of detail. Okay, so what about B lymphocytes? So again, B lymphocytes become activated, and then they differentiate into different subsets: the memory cells and your plasmablast cells, which are important for making antibodies. So these are real biosynthetic machines here, churning out antibodies. So you can imagine metabolism is going to be important for these cells. So again, it's a similar story to the T cells. Upon activation, you get very clear increase in metabolic rates. In the memory cells, these then become reduced a bit, whereas these plasma cells, which continue the high biosynthetic outputs of making lots of antibodies, these cells maintain these elevated levels of metabolism. So if you take a look at the general picture, so it's quite similar here. They have high levels of glycolysis, aerobic glycolysis. They increase their metabolic machinery in the mitochondria. Glutamine is also an important substrate in B cells, and they do engage the TCA cycle. So it looks quite similar, and makes sense based on the fact that they are engaging in biosynthesis. There's also evidence that the B cells are using the pentose phosphate pathway, and this is important for maintaining the redox status. People have looked at de novo lipid synthesis in B cells, and that's also really important. So they have a very metabolic configuration that's very consistent with a really highly metabolically active cell. One interesting part that's been found to be crucial for B cells, which really links to their sort of unique functions, is the fact that the hexosamine pathway is really important for B cells. Now, we haven't discussed the hexosamine pathway, but it again branches off glycolysis at the level of glucose-6-phosphate, and it's important for making this molecule here, UDP glutenac. So glucose and glutamine combine to make this UDP glutenac, and this is a really important molecule for antibody glycosylation. Now, all antibodies are glycosylated, and glycosylation is really important for the antibody function. So here's a picture of an antibody, and here is the hinge region of the antibody. Here's the variable regions, and here's the constant region, and this is a very nice review if you're interested in it. But the glycosylation pattern here within this hinge region is essential for the function of the antibodies, especially in terms of their ability to recognize FC receptors. So this is, remember, the FC region of your antibody is how it communicates with the rest of the immune system in terms of what receptors it binds to. So, and this glycosylation is really important for that. So for that reason, the glucosamine pathway is very important to allow this glycosylation to happen. So in terms of metabolic regulators, what are the key metabolic regulators in B cells? So again, we see a lot of the usual suspects that we've seen previously. HIF1 alpha is important for B cell development. It's also important that during this activated stage. c-Myc is also important. HIF1 alpha also is important in this long-lived stage, kind of similar to what we saw for the cytotoxic T cells. And of course, mTOR seems to be, which is a global metabolic regulator and seems to crop up in a lot of cells, is very important here as well. So there's this very nice review you can have a look at if you want more information about that, discussing all these metabolic regulators and metabolic fluxes during the different stages of B cell development, from the bone marrow to peripheral activation and a terminal differentiation. And of course, we now think about these as key metabolic regulators, but before we were even focusing on metabolism, we already knew that some of these signaling molecules are already important for the function of lymphocytes. So we knew that mTOR was really important for T cell and B cell functions before we were even considering the fact that it's a metabolic regulator. So research had already found out that mTORs are critical for differentiation of B cells and T cells. Before we were thinking of it, before we even thought that that's because it's controlling metabolism. So I guess we came to understanding metabolism of immune cells a little bit late, but we're certainly catching up now. So, what about direct metabolic perturbation? What if we start playing around with these metabolic pathways? How does this affect this pathway of B cell activation and differentiation? So people have looked in the GLUT1 knockouts. So GLUT1, which is also called SLC2A1, is one of the glucose transporters. It's not the only one, so lymphocytes also express GLUT3 a lot, and that tends to be the sort of basal glucose uptake. So it's not, the level of GLUT3 don't change much. But GLUT1 is a transporter that's robustly increased upon activation. And if you knock out GLUT1, you fail to generate these mature B cells. You have a deficit in the production of IgM and IgG responses. So the whole T cell B cell response is impaired. If you add things like direct inhibitors of glycolysis, this isn't a very elegant way of doing it. It's a bit of a dirty experiment, but nonetheless, it's informative. So adding 2DG, which blocks glycolysis, it also blocks other pathways like pentose phosphate pathway and hexosamine pathway. But nonetheless, the germinal center doesn't form properly; it collapses. So it's clear that glycolysis is important, or certainly glucose-dependent pathways. Similarly, if you add oligomycin, which inhibits your respiration, or oxphos, you can't form a germinal center, and you have a failure for class switching to IgG1. So very clear, emerging evidence, and more is coming out day by day. It's very clear that metabolism is also important for B cell differentiation and function. Another experiment, so let's not forget other fuels like glutamine. Glutamine deprivation also affects B cell differentiation, activation, and class switching and proliferation. So, as I said previously in the previous slide, glutamine seems to be a very important fuel for B cells, certainly more so than NK cells, where it really is a rather minor fuel. Okay, so I hope I've brought you through these three lymphocytes populations, some in more detail than others. So I talk about NK cells most, but I guess because we've worked on that most, and I can show you some primary data. But the take-home message really is that cellular metabolism is, well, it's different in all these subsets, but it's also integral to all the responses of these lymphocytes. And so we really need to consider the metabolic configuration of these cells, and the metabolic regulators are important. And through understanding that, I think we will find new ways to manipulate metabolism therapeutically to be able to control these responses in the context of cancer therapy, or in the context of treating patients with autoimmunity, to give us new avenues for therapeutics beyond what we already have. So I think it's quite an exciting time to be working in this field, as new strategies for controlling immune response are emerging all the time from this immunometabolism field. Okay, so that's the end of this lecture. In the final lecture, we're going to talk about how our understanding of the metabolic configuration of these cells has implication for why these cells don't seem to be working in certain immune microenvironments, such as why lymphocytes tend to be dysfunctional in cancer, etc. So thanks for your attention, and take care.